Hello and welcome everyone. In this podcast we're going to talk about the enzymes that methylate DNA. So we've talked a lot about the impacts of DNA methylation. We've talked about the importance of DNA methylation, but now we want to talk about specifically those enzymes that do the job of methylating DNA. Let me list them here. DNMT1, DNMT2, DNMT3A, and DNMT3B. And I'm going to immediately discuss one of them so we can get rid of it. And that's this one here. Your book mentions it briefly and correctly labels it as a tRNA methyltransferase. It was at one time, because of its sequence, was thought to be a DNA methyltransferase like these other three, but we now know that not to be the case. It's still a very important enzyme in methylating tRNA, but not really part of our discussion in these podcasts, so we'll get rid of it. So to remind you, the DNMT of these enzymes stands for DNA methyltransferase. They take up methyl and they enzymatically transfer it to a cytosine. So they all have the same general function, and that is to make methylated DNA. However, they do have some differences that I want to point out real quick here. The main difference between these three is that DNMT1, it functions to maintain methylated DNA, whereas DNMT3A and B both generate methylated DNA in a de novo manner. Okay, first I'd like to talk about de novo methylation. This requires several proteins, but I'm going to talk about three of them right now. The first, as I mentioned before, were the first two, DNMT3A, DNMT3B, and then this third protein called DNMT3L. DNMT3L is not a methyltransferase, but it is a regulating factor in methylation. And we know it's important because, I'll just put it right here, DNMT3L mutants have the same phenotypes as DNMT3A or B mutants. So we know it's important because of that. We also know that it interacts, this 3L subunit interacts with 3A and 3B. And we know that because if we take a cell that has 3A, 3B, and 3L in it, these three proteins, and we crack open the cell, and in cracking the cell open, we take an antibody. So antibodies are really useful for this, that is specific to 3A, so antibody to 3A. So it grabs all the 3A in the cell, and anything that's attached to 3A comes with it. And so whenever we pull 3A out of this cell, we also get 3B and 3L. The more technical way of saying this is that 3A and B co-IP for co-immunoprecipitate with 3L. They're together. You get one, you get them all. So these are the proteins that are involved. But what do we mean by de novo methylation, and why is it important? Essentially what we mean by this is, by de novo, is that you have a blank slate, so to speak. You have this DNA sequence here with no methyl groups on it at all. There are cytosines throughout this sequence, but no methyl groups. But there are specific genes here in this network I just drew here that need to be methylated. But there's no template to use, so it has to do it by a different mechanism. And so these proteins here, 3A, 3B, and 3L, will selectively methylate these cytosines. And not all of them, just the ones that need to be methylated. Now this is very important during certain stages of life. And the most important is during embryonic development. As an early embryo, most of the methylation has been removed. Yet some of these cytosines need to be methylated early, and in many cases need to maintain methylation later on in life. Now, how it knows which cytosines to methylate is a different process that we're not going to get into right now. But the take home is that these three proteins work together to create a methylation pattern in the embryo without using any template. It makes them from scratch, so to speak. Now let's counter that with what DNMT1 does. And what DNMT1 does is it maintains methylation. Once the embryo has established the methylation pattern, it's important to maintain it, if indeed adding those methyl groups were important. So it's important to maintain it. And I think the best way to explain this is to look at a DNA sequence here. Well, a partial DNA sequence. And so let's say we have a C here and a G here. C and G's bind together, and we have A's. We'll just draw a few things here. G's, G's, T, C. And they're going to have the complementary bases over here. So this is T, A, C, C, A, G. And for the sake of this discussion, let's just say all the cytosines have a methyl group on them. Now, when this cell divides, the DNA is replicated. And this strand here is used as a template to make the new double helix. And this strand is used as a template. So let's go ahead and draw our two new cells. So this was our original one. These will be our new ones. And I'm going to draw the new DNA strand, like so. I'm going to draw the purple ones first, our parental ones. And if you remember from genetics, we call this semi-conservative replication, because the new cells maintain half, the word for semi, half of the genetic material, and they generate the other half. So it conserves half of it for each cell. Okay, a T and then a C. And remember, well, let me go ahead and write over here now. G, T, A, C, C, A, G. And remember, these have their methyl groups on them, the C's, like so. Now the new DNA that's just been made, let's draw its complementary bases. And what you'll notice is that the new DNA that's just been made, and we have the cytosines here and here, and the original parent, those were methylated. But as DNA is being replicated, it only brings in the standard bases, guanine, thymine, adenines, and cytosines. It doesn't bring in a methylated cytosine. So in order to maintain this methylation pattern, DNMT1 has to re-add the methyl groups here. So its job, as you can predict based on what I wrote up here, is it maintains the original methylation pattern. If in one cell in the pancreas you want a gene to be turned off because of methylation, when that cell divides, you want that same repression, the same repressive mechanism stimulated by the methyl groups to be maintained. And that's the job of DNMT1. Now, how does DNMT1 do this and the other DNMTs don't? Well, DNMT1 has the feature of being able to preferentially bind hemimethylated double-stranded DS DNA. So before I drew these new methyl groups here, it prefers to bind DNA like this that only has methyl groups on one side of it. DNMT3A and 3B prefers to bind DNA that has no methyl groups on it. Now, what would happen if we mutated DNMT1 and we weren't able to maintain these methyl groups? So remember, these methyl groups are established as an embryo, and even as a young embryo, DNMT1 takes over because every time that embryo cells divide, it needs to create the same methylation pattern. But if you don't have DNMT1 around to remethylate these cytosines, two things happen. The first, which shouldn't be too surprising, is that methylation is decreased. The second thing we observe, which gets to the point of why this is really important, is the embryo dies. It's able to set up the original Denovo methylation pattern because DNMT3A and B and L are there, but if you can't maintain it with DNMT1, the embryo dies. Okay, in the last part of this podcast, I want to talk about DNMT activity. How is it regulated? And if we think about how any protein or enzyme is regulated, we can think of multiple ways. So as we think about how DNMT itself is regulated, let's remind ourselves that DNMT, in this case DNMT1, is a gene itself. And as a gene, it needs to be regulated. Granted, its job is to help regulate other genes, but at some point it has to be determined whether or not DNMT1 itself is going to be turned on or off. And like any gene, DNMT is transcribed into a messenger RNA that has a series of exons and introns. This messenger RNA is then processed, where a G cap is added to the end, the exons are maintained, introns are lost, and we add that poly A tail to the 3' end. This processed messenger RNA then is translated into the DNMT1 protein. Throughout this whole process, there are many ways that DNMT1 can be regulated. And I'm going to talk about a few of them. And the first is at the level of transcription. And all I want to say about this is that, and all I want to say about how DNMT is regulated at the level of transcription is that there is this transcription factor called STAT3. STAT3 is an important transcription factor in that it is linked to cell proliferation, inhibiting programmed cell death, and angiogenesis. Remember, some cells we want to go through programmed cell death, other cells we want programmed cell death to be inhibited. Some cells, or at some times of the cell's life, we want it to proliferate, other times we don't. Angiogenesis is a very unique pathway, and that is the pathway of building blood vessels. So during embryonic development it's very important, and during wound repair it's important to build blood vessels to those sites. All of these are bad news when it comes to cancer, because cancer is linked to an overcell proliferation, the inability to inhibit programmed cell death, and increase angiogenesis. So this is linked to cancer, but we also know that it's linked to activating DNMT1. Again, this will bring us back later in the semester to how methylation and epigenetics as a whole is linked, or can be linked, to cancer. The next pathway that DNMT1 is regulated is at the processing step. I'm going to come back to this because I want to spend a little bit more time talking about that. The last one that I want to mention is at the enzyme level. Your book goes into great detail on how various intermediates in other pathways are linked to the activity of this enzyme. The spermine and spermidine and other polyamines. I don't want you to know that. It's an interesting story, but that's not what I want you to know. What I do want you to know, because this is an important biochemical regulation pathway, is that when cytosine is converted into 5-methyl cytosine, it creates as a byproduct ADOHCY. ADOHCY inhibits DNMT1, and it does so in a competitive manner. So I'm going to write competitive inhibition. And I think it's worth mentioning what that means. So for instance, if we go to the enzyme drawn here, and let's give it an active site now. Let's say it has this active site right here. And let's say the preferred substrate is cytosine. It has a higher affinity for cytosine. However, as more and more 5-methylcytosine is generated, this byproduct here, ADOHCY, accumulates. And at high concentrations, this AH, as I'm going to abbreviate it, has a preference for this binding site over cytosine. And if it now has a preference for this active site, cytosine cannot get in there. So this is one of the mechanisms that DNA methylation can be regulated, or rather DNA methyltransferase can be regulated by a competitive inhibition of a byproduct. Okay, I want to talk a little bit more about how DNMT1 is regulated at this processing level. More specific than just processing, I want to talk about how DNMT is regulated by alternative splicing. And this DNMT gene is a fairly long gene, and so I'm just going to draw kind of a brief diagram of it. There's more introns and exons in it than I'm showing here. I'm just drawing this for simplicity. So what we know is that this one DNMT1 mRNA can be alternatively spliced to produce 3 different mature messenger RNAs. And we're just going to draw some as an example here. Let's say this one here contained E1, E2, E4, and E5. And this one here contained E2, E3, E4, and E5. And this one contained all of them. And so why do we have these 3 different forms? Well, we know one of them is expressed in the spermatocyte at a stage when crossing over is occurring. This one, let's say this one is produced in the oocyte. And I'm going to come back to that one in just a moment. And this one over here is produced in somatic cells. Remember, the job of DNMT1 is to maintain the methylation pattern. So once it's been set early in embryonic development, it has to maintain that methylation pattern, and that's why we need it here. So why is it we need these 3 different forms of DNMT1? Well, I'm going to start here with somatic cells. We know we need a form in somatic cells that's important in maintaining that methylation pattern that we've talked about earlier. And so that's what it's doing here. So why do we need these different forms in spermatocytes and oocytes? Well, the first point pertains to both of them, and that has to do with crossing over. During DNA crossing over events, during meiosis, DNMT1 is shifted to the cytoplasm. This is very important because we know that during crossing over, the DNA is at a very vulnerable state. Because we know if the DNMTs were in the nucleus at that time of crossing over, that methylation would occur at most cytosines. So it shifts to the cytoplasm to prevent random methylation. So the next point I want to make is that DNMT stays in large reservoirs in the cytoplasm. So you have the oocyte here with the nucleus, and the DNMT forms these large pockets of concentrated DNMT in the cytoplasm. We don't know really why, but if you think about when this egg gets eventually fertilized and starts to grow as a young embryo, it's going to divide really fast, because that's what embryonic cells do. They divide really fast. Maybe this DNMT that is in the cytoplasm after fertilization rushes into the nucleus to maintain that established methylation pattern, so that during those early rapid rounds of cell division, that methylation pattern isn't lost. Okay, that's all I have to say for this podcast. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.